Wow. Applause before the talk. I can't remember the last time that happened. I really want to thank Alfie for inviting me to talk to you all today. This is one of my favorite subjects to talk about, and like he said, I've been involved in it a long time, and you're going to get some idea of that, I think, soon. I'm even going to tell you what somatic embryogenesis is and show you photos of it. It's not too hard to understand. It's just a big word is all it is. And I think you'll enjoy what it does. So I got to start off with a few questions. One is, how many of y'all have ever seen an American chestnut tree in person? 2. Okay, let's go a little further. How many of y'all have come from a town that has a chestnut street or road in it? A few more. Okay. If you were from up north, you probably, all those towns have a chestnut street. Mine had 2. I don't know why. It did, though. Okay, last question. How many of y'all have ever eaten a chestnut of any kind? Okay, that's pretty typical. Okay, so I'm going to tell you where you can, if you ever want to try a chestnut, where you can pick up some. It's not that hard to find, especially this time of year. Actually, if you go to Publix or Kroger, probably starting about mid-November, you can buy them. They'll have them there in the fruit and vegetable section. They'll probably have a basket or a bin of them if you want to try them. But I'm going to tell you a little bit more about those. So often, when I used to start a talk about my lab's work, I would just show this photo and I wouldn't tell people what it was. I'd ask, well, what do you think these trees are? And people would look at that and they go, oh, those are redwoods in California or giant sequoias. They ain't, as we say in the South. These were American chestnut trees over 100 years ago. This photo was taken, as you can see at the bottom, in North Carolina, in the Smoky Mountains, right around 1900. Those guys at the bottom there that you see, every tree you see in there is American chestnut. They're just posing for photos. I thought they were loggers, and they took this photo just before these trees were cut because a lot of American chestnut was cut down, and you'll see why soon. But no, they weren't. These trees lived for a while longer, and you'll see what happened to them, or you'll hear what happened to them. So you may have seen leaves that look like this on some trees on campus, because we have planted on campus as an ornamental an oak tree that's related to this, because they're all in the same family. The oak family has the oaks, the chestnuts, and the beeches. And those elongated leaves with serrated margins, with little hooked teeth on them, you'll see trees like that, but they're actually something called sawtooth oak. They look very similar to chestnut. It's about as close as you're going to see it. We do have some Asian chestnuts on campus. The last one that I know of that's left is actually in the tennis stadium, way down on South Campus. It's got a nice deck around it. But it's a Chinese chestnut tree. You can see the leaves. That thing in the middle is the fruit, and it's actually a spiny burr. You'll see some photos of it, and inside are 3 nuts. And those are the chestnuts that the tree makes. This is often shown as the original range of the American chestnut tree in North America. You can see it goes all the way up into New England, a little bit of Canada, and extends down through the Appalachians all the way into, it looks like extreme North Georgia and North Alabama, all the way over to Mississippi. This is actually wrong. The person who drew this map, and this map is everywhere, and other ones have the same range, was made too late. This is where chestnut was about the time blight came through in the 1900s, in the early 1900s. But, and I don't have time to talk about this, there used to be chestnut, and you'll see it in this map, all the way down onto the coastal plain in Piedmont of Georgia and other southern states. This map is a really interesting map. It was made by a fellow named Nathan Klaus, who works for the Georgia Department of Natural Resources. And what he did was really, he's a very smart guy. He wanted to know where there was chestnut back in the 1800s, like 1830. So what he did was this. You may or may not know that there were several land lotteries in Georgia where the state took land, sometimes from the Cherokees or other Native Americans, got rid of them, and then sold off the land in parcels. Actually, sometimes they gave it away. The parcels were different sizes, maybe 60 acres, maybe 150 acres. And when they mapped those parcels before they distributed them, they marked the corners of each parcel by naming the trees that they found, the biggest tree they could find near the corner of that parcel. Nathan found these old maps, and every place where someone put American chestnut, because these maps, these land lotteries were back in the early 1800s, I think the last one was maybe in 1840. And wherever he found a chestnut, he marked that spot. And all the yellow you see there and the green, that's where chestnut was at one time. Unfortunately, all of the chestnuts outside of the mountains were killed by a disease called Phytophthora root rot, probably brought in by people in Savannah who brought in azaleas and other ornamentals from Asia carrying that disease organism. And it wiped out, and it's still a problem, it wiped out all the trees at the lower elevations because it was a warm weather pathogen and it didn't get up into the mountains. It was too cold up there. And that's why that map showed just the trees were only in the mountains, but they weren't. Jimmy Carter writes about that he had American chestnut trees on his family farm when he was growing up in Plains. So there were chestnut trees that extended a lot further than just the mountains. The green areas are where the trees were the densest in Georgia. You can see there, it's essentially north of Atlanta, and then on up into White County, Raven County, all the way up into the line into Tennessee and North Carolina. So there were a lot of trees. So these trees were incredibly useful, and they were useful in lots of ways. First, we can talk a little bit about the wood. The wood of American chestnut was unlike the wood of almost any other tree in North America, except for maybe black locust, in that it was very high in tannin content. In fact, the bark and the wood were very high in tannins, tannic acid. And what that did is when you use the wood for outdoor purposes, and I'll show you a slide in a minute, to build anything outside, whether it was houses, barns, telephone poles, railroad ties, that meant that it was very durable, and by durable I mean rot resistant. The organisms, the fungal organisms that would rot the wood couldn't attack it because the tannins would repel them or kill them. So essentially what they use pressure treated lumber for now, that's what they used American chestnut for back then. All kinds of things were made out of American chestnut, but anything you wanted to stay outside in contact with the ground for decades, and you'll see maybe even hundreds of years, was made out of chestnut in the eastern United States. Okay? This is wood. This is actually after blight went through, and there were lots of dead trees. This was actually salvaged wood. They cut down all the dead trees and used the wood because the wood was still sound. In fact, they're harder to find now, old cabins and barns up in the Appalachian Mountains that were built 150 years ago. The timbers that hold them up are still just as sound as they were when those buildings were built all those years ago because the wood is so durable. This is probably the best illustration of how important American chestnut wood was to the people of the Appalachians. Everything that you see that's made of wood in this photo is American chestnut. The shingles, split shingles on the house, the siding on the house, all the buildings, the sidings on all the buildings, it's all American chestnut. The timbers holding up the house, those are American chestnut. The fence is chestnut. Even the wood that these ladies are heating their washing water with was American chestnut. This was a central wood, especially in the southern Appalachians, for people to use. They depended on that wood for anything that had to stand up to outdoor conditions. There was a lot of furniture made of chestnut wood. It was not a fine furniture wood like black cherry or black walnut, but it was an everyday kind of a furniture wood, and it was often said it was often called the cradle to grave tree because if you were born in the Appalachians before 1900, it's likely that when you were born you were rocked in a chestnut cradle, and when you were put in the ground it was in a chestnut coffin. It was used for everything. The picture on the right is actually a piece of furniture in my house about 30-something years ago. I went to an antique store here in Athens, and I saw this, and I went, oh, that looks like chestnut. And sure enough, it's American chestnut. I think we bought it for about $350. It's harder and harder to find furniture made of chestnut. There was tons of it at one time, but now they're highly valued antiques. So I don't know what this would cost now, but probably closer to $800,000. And it's not even a really great chest, but it is kind of a rare thing to have. How about the nuts? So the chestnuts were very different from all the other nuts, and any nut you've ever tasted in the eastern United States. Chestnuts are different from pecans and walnuts. When you eat a pecan or a walnut, they have fat. It's good fat, but their storage product that you're eating is fat. Chestnuts have very little fat. Their storage product is starch. So a lot of people, when they pop a chestnut into their mouth for the first time and bite down on it, they go, "Ugh, this doesn't taste like a nut." But it kind of grows on you. They're sweet, and they're a little mealy. You roast them. You can eat them raw, but they don't taste like what you think. But after a while, you kind of get it, and they kind of grow on you, and they're really quite nice to eat. So chestnuts, I want to tell you couple other things about them. You can see how important they were to wildlife. It's said that wildlife were more abundant and grew bigger when the chestnuts were still here. Why was that? Chestnuts are unlike the oaks and hickories, which is what these animals have to eat now, acorns and hickory nuts, in that every single year was a great crop of chestnuts that came off the trees. And why was that the case? It's because they flowered later than the oaks or the hickories. Often, there's a late freeze, and if the flowers on those trees freeze, no acorns, no hickory nuts. Chestnut doesn't flower until like May. It's very rare to have a freeze later than that. So their flowers didn't get killed. They always had a good crop of chestnuts. So you can see all the wildlife, the bears, the deer, the turkey, everyone depended on that. People depended on chestnuts too. In the southern Appalachians, people would do two things that were very important to them. One, they would go out and gather chestnuts in the fall and bring them in and then sell them at a local store which would then ship them up the railroad up north, because they would sell roasted chestnuts on every corner in New York City in the 1800s and early 1900s. And they needed more chestnuts. So they could make, even during the Depression, they could sell a bushel of chestnuts for $5, which was enough to buy a pair of shoes. The other thing that was probably more important to them was everyone had hogs in the Appalachians. They all kept hogs so they could sell them. And so they would drive their hogs up into the mountains in the fall to eat chestnuts and fatten up so that they could sell them for more money when they were ready to sell. Now, people in the cities ate chestnuts too, as I just said, and there were chestnut trees in the city. The engraving that you see there on the left is in Fairmount Park in Philadelphia. And what these people are doing is something called chestnutting. In the fall, people would go out into the parks with baskets. They'd get some folks to climb the trees and jump up and down on the branches, which is what that guy's doing, and it would rain down chestnuts. They'd pick them up, take them home, roast them, and eat them, use them in dishes they were making. So chestnut nuts were very, very important. All right, this is the last use slide I'm going to show you. What are these old boys doing? As we say in Georgia. Did you hear that? Moonshining, right. So American chestnut was the wood of choice for moonshiners because you could burn it with almost no smoke. Other woods make quite a lot of smoke. If you had an illegal still back in the mountains and you didn't want the federal agents to find it by, oh, there's some smoke rising back in there, we know where there's a still, you would use chestnut to heat your still. I forgot to tell you one other thing about chestnut that's really wood, and it just reminded me. That's really important. When the first settlers got here, they didn't have sawmills, and many of them didn't even have saws. How did they make planks to build their houses with? Boards. With chestnut, the grain was so straight that they could use a splitting maul or a hammer and wedges and make their planks that way. Other woods are difficult to do that with because they weren't as straight grained as chestnut. You needed a sawmill or at least a saw to make your boards. So there's another thing that was very important about the tree. All right, now we get to the nasty part. So in 1904, there was a forester working in what is now the Bronx Zoo. It's the New York Zoological Park in New York City. His name was Merkle, but he was an M-E-R-K-E-L Merkle. I'm an L-E Merkle, no relation. He noticed that the chestnut trees along the streets in the zoo were dying. And now apparently he was one of the first people to notice it. He checked out the trees and he noticed that there were these cankers on the sides of them, and he decided to take a little sample out of one. He scraped a sample out of one, went over to the New York botanical garden where there was a plant pathologist and asked this guy, told him, these trees are dying, I got this stuff, what is it? And the pathologist, his name was Merkle, he looked at it and decided that it was a pathogenic fungus. He named it Endothea parasitica. And that is the chestnut blight fungus. You see now, the name is not Endothea parasitica. About 25 years ago, for some reason, the taxonomists changed the name to Cryphonectria parasitica, which is the current name of the fungus. How did it get here? It got here because it was native to Asia. In Asia, it grew on the Asian chestnut trees, Chinese chestnut, Japanese chestnut. Because it co-evolved, these trees co-evolved with the blight fungus, they would get little cankers on them, but it didn't kill them, right? People in the United States, they didn't want to have to climb huge canopy American chestnut trees to get the nuts. And they also knew that the nuts on the Asian trees were bigger. So they brought over Chinese chestnuts and Japanese chestnuts to plant in their yards or in their orchards. And they carried the chestnut blight fungus with them. It got off of those trees onto our American trees. American chestnuts had never been exposed to chestnut blight fungus before, and it went through them like a hot knife through butter. Remember what happened to the Native Americans when the Europeans got here? Smallpox, other diseases killed sometimes 90% of Native American tribes. That's the same thing that happened with American chestnut, but the devastation was almost total. What you see here is when you see an infected tree, you often see these yellowish or even orange pustules on it. Those are the sporangia, the spores by which the fungus spreads from tree to tree, either by wind or by splashing water. Birds will sometimes, woodpeckers will land in it, get it on their feet and carry it to other trees. It spread very rapidly. The photo on the right shows the sunken, killing canker that once a tree gets this, what happens is, and I'll show you another slide in a minute, the fungus fans out underneath the bark. It'll enter through any wound in the bark, maybe a broken branch, and then it'll start growing out and it'll grow under the bark and it will kill the living tissue under the bark. That's the phloem and the xylem, cells that carry water up the tree and sugar down the tree, and the cells that make the phloem and the xylem. And once it kills a complete ring around the tree, water can't move up the tree, sugar can't move down, the tree dies. Sometimes within a year or two of getting infected. And that's what the sunken killing canker does. Now, how does it kill the tissue? This is a weird fungus, not that weird. It's called a necrotrophic fungus in that it kills the tissue of the plant it's attacking, which is almost always a woody plant, and then eats the dead tissue. Okay? How does it kill it? It secretes a chemical called oxalic acid. Remember that name, oxalic acid, because it'll become important later. You can actually grow this fungus in culture, in a petri dish, and it makes the same orange looking pustules in there. And the drawing on the right shows the mycelia getting under the bark and forming the wedge that eventually kills the tree. So what happened? From New York City, the fungus spread at the rate of 200 miles every 10 years down through the Appalachians, hitting Georgia about 1940, killing all the big trees in its path. It was probably about 4 billion trees that were killed. It was the most devastating forest tree disease ever recorded in history. And I think, the map kind of tells it all there, how it did it. Here's some scenes of what it looked like, which are pretty depressing. That's a dead snag there. The guy's actually inside the dead tree. That was a huge chestnut tree. A whole mountainside full of dead trees on the upper right. And then this is a picture of what people called ghost forests. What would happen is these trees would die from blight, but because the wood was so decay resistant, the tree would just keep standing there. The dead tree, the snag would keep standing there. The bark would fall off, and you were left with these white trees standing there, sometimes for years and years before they eventually fell over. They were called ghost forests. People in the Appalachians, you can imagine how devastated they were when they saw all their chestnut trees dying. There's no one hardly left alive today that really remembers the chestnut blight coming through. But a lot of these people were interviewed, and there are interviews you can find on the web of these people, and they talk about trying to sleep at night, and suddenly they hear this gigantic crash because the chestnut tree, maybe 20 yards away, had come down in the middle of the night. No more nuts, no more wood. It made a huge negative impact on their lives. But you can still go up in the mountains, even the Georgia mountains, and you can find chestnut trees. They look like shrubs now. Why is that? For some reason, and it has never been determined, and I've been to a lot of meetings where it's been discussed, the blight kills the stem, but it does not kill the root system or the stump. The transition is called the root collar, right where the roots meet the stem. What happens is there's all kinds of buds sitting there in the root collar, and when that main stem gets killed back, it allows them to sprout, and they grow. And sometimes they will grow for a long time, maybe 20 years, and maybe get 15, 20 feet tall, maybe even big enough to make some nuts. But eventually, they get reinfected, they get killed back to the ground. And so the trees go through these continuous cycles of dieback and resprouting. Most of them are about this size, but once in a while you find some relatively big ones. I'm going to show you some big ones that are a little different too. So what did people try to do about this? And this is like all the way from the very beginning to today. All these approaches to trying to combat chestnut blight and restore the trees. Of course, what they did in New York City first was they tried spraying fungicides on the trees and pruning off infected branches. It didn't work because, of course, the fungus was underneath the bark where it was protected. So that didn't save anything. There's been whole books, or parts of books anyway, written about efforts by some states to use quarantine. What Pennsylvania did is they knew the blight was coming down from New York, so they hired all these loggers to go out and cut down every chestnut tree, living and dead, diseased or not, for big swaths of Pennsylvania. Sometimes, like a 50-mile, they wanted to cut a 50-mile wide barrier, and they thought, oh, the fungus can't get over that, but it didn't work. In fact, they actually made it worse because these loggers, they would carry their tools that they'd use to cut down infected trees over and start cutting down the nice, healthy trees and spread the blight that way. So and they spent millions of dollars on this when $1 million was a lot of money. Completely failed. And a few other states like Virginia tried quarantines as well. It didn't work. They looked for natural resistance to the blight fungus in American trees. Remember, 10% of Native Americans were resistant to smallpox. How about 10% of American chestnuts being resistant to blight? That was not the case, although I'll show you there are some what are called large surviving Americans. That's what LSA means. These are trees that I'm going to show you pictures of in just a few minutes that were probably saplings when the blight went through, but they have continued to live. They get cankers on them. They look sometimes not that great, but they have some kind of resistance because they're surviving. And they say they have some pretty good resistance. It's not published very much, though. There's not much to find out there about it. I've worked with all of these trees, that is nuts that they've sent me off of the Thompson Ragged Mountain, the Amherst tree in Amherst, Virginia, and this one I don't know if they work with or not. This is in Kentucky. It's called the Adair tree. And we have cultures that I'll show you that come from these trees. This is the other breeding program that I'm heavily involved with the American Chestnut Foundation. It's abbreviated TACF. Someone already had ACF. It stood for something else. So they decided to capitalize the T, and they call themselves the TACF. Here's the breeding program with the Asian chestnuts. And this is called a hybrid backcross breeding program. It's pretty easy to understand if you look at it a few times. So we want to get the blight resistance genes from Chinese chestnut into American chestnut, right? But here's what Chinese chestnut trees look like. They're low branching. They are not canopy trees. They're orchard trees, although there are some bigger ones and straighter ones. Well, you want to get the blight resistance genes into American chestnut, but not the other Chinese chestnut genes that you don't want in there. So how do you do that? First, you make a hybrid between the two. It's called an F1 hybrid, and roughly 50% of the genes in that hybrid are American, 50% are from the Chinese chestnut. Then you want to start getting the Chinese chestnut genes you don't want back out of it. So what you do is you cross that to an American tree, and those progeny now have 3/4 American chestnut genes and only 1/4 Chinese. At that point, you take some blight fungus and you inoculate those trees, and the ones that look kind of like American chestnuts but have some blight resistance from Chinese chestnut, those stay in the program, and you back cross those again to another American tree, and so on. And every time you back cross to an American tree, you inoculate to test for resistance, and you decrease the amount of Chinese chestnut genes that are still there until by what's called the back cross 3 generation, you have trees that are 15/16 American 1/16 Chinese, and supposedly blight resistant. And then you intercross those trees to bring the blight resistance genes into the homozygous state, so you have 2 copies of each resistance gene. And by around 2000, they actually had these, what are called the BC3F3 generation. And they released these to people to grow. They put them out in tests, and they didn't perform well. It was kind of disappointing. What happened? Well, mistakes were made, as we say, in the breeding program. And I know the breeder really well. He's a very good guy, and he was carrying a huge load on his shoulders. 2 things happened. One is they missed some of the resistance genes that were in Chinese chestnut. The breeding program was started actually by this guy. Instead of doing his own F1s, he went and got some BC ones that someone else had already done. And so he missed some of the genes that he should have gotten from Chinese chestnut. There's other things that happen too. I'm not going to go into it. But essentially, when they test these trees now, they pretty much have the same amount of blight resistance as the original F1 hybrid, not what they wanted, okay? So there are some trees, though, at these various stages, though, that look very nice, and I'm going to show you one of them. Oh, in a minute. Their breeding program is in Southwest Virginia at Meadowview. It's not too far from Virginia Tech. And they have the trees where they do the breeding. This guy's up in a tree. You see the pollination bags here, where they're pollinating the female flowers with pollen from other trees. And sometimes you'll get up in a cherry picker to get up in the trees. And then these are all outplantings of their trees there where they're testing them. They tested thousands and thousands of trees. This is one. It turns out that each state has its own chapter. Not each state, but most eastern states have their own chapter of the American Chestnut Foundation. And Penn State had a really great program in their chapter. This is a BC3F2. That's the stage before the last one. Looks like an American chestnut tree. It's been inoculated twice with 2 different strains of the fungus, still growing. It gets a canker on it, but there's some resistance there. And the Pennsylvania chapter has a couple of trees like this. I took that photo when I visited there for a meeting. Georgia has its own chapter of the American Chestnut Foundation. It was started much later than the Pennsylvania chapter. It's about 15 years old now. And I'm a member, of course. And I'll go ahead and make a pitch for it. They need young people. It's all old farts like me that are in the group, and we can't climb ladders and stuff like that. Well, we can, but we fall off. And so they need young people that will go out and help with the breeding and planting and testing and all that stuff. If you have any interest in this, it's easy enough to find them. Just put in American Chestnut Foundation on your browser, and it'll take you to the national, and then they have tabs for all the chapters. And we meet once a year in the spring, and we have some pretty good scientists that kind of are in charge of the breeding and stuff. And it's a lot of fun. It's a nice group of folks. We do need more people, and especially young people. All right, so let's get into the last part here, and this is the genetic engineering part, okay? So, the original plan, the breeding program, regular conventional breeding, hasn't come out that great so far. Well, about 1990, actually I started in 89, people started thinking about, well, why don't we just find some blight resistance genes and genetically engineer them into American chestnuts, and then we don't have to do all this back cross breeding and stuff. So I'm sure you all know this already, but I'm going to tell you anyway. If you eat anything that's made with soybeans or corn, and it's not labeled no GMOs, you're eating genetically engineered stuff, right? You all know this, right? Okay. Anyone grown 3 arms yet or anything? Not yet. Okay, that's good. So, and cotton, also cotton is all genetically engineered, mostly for the plants to be insect resistant. That's what most of the genetic engineering is, right? But it does come through. The genes are in the products that come out. And these are all synonyms for the same thing: genetic engineering, gene transfer, genetic transformation, recombinant DNA, GMOs. Well, you might not know how it's actually done. There's different ways to do it. The easiest one, which we've used some, is a gene gun. Essentially, you take tiny little metal particles, coat them with the DNA, and blast them right into the cells. Believe it or not, it actually works. But you get some crazy products out of it. Like sometimes you'll get a plant that has 50 copies of the gene, and you don't really want that because they all suppress each other. What's usually done, this is by big ag companies, by universities, by everybody, is they use a little bacterium. It's a soil bacterium that's actually a natural genetic engineer. It's called Agrobacterium tumefaciens, and it causes diseases in plants and in trees diseases called crown gall. And so what it does This is a picture, I should have labeled these pictures. This is a picture of a plant cell with little agrobacterium stuck to the outside of it. And what they're doing is they're actually injecting a little circle of DNA into the plant cell. It's their DNA, it goes into the plant cell, and it actually gets incorporated into a chromosome in the nucleus. And that's what causes this crown gall, because on that circle of DNA are genes that say, make food for the bacterium. And so it makes food for the agrobacterium, and it says, make more cells so it'll make even more food. So it makes a gall, okay? So it's a disease, and it's nasty looking, right? It's called crown gall because that place where the stem meets the root, that's called the root collar or the crown, root crown. That's why it's called crown gall. Trees get it, other plants get it. About 1980, scientists figured out, much smarter scientists than me, figured out that they could cut those disease genes out of the plasmid and stick in whatever gene they wanted into that circle of DNA and have agrobacterium put that inside the plant cells. Okay? Maybe it's a gene for disease resistance. Maybe it's a gene for faster growth. That's how they do it. So I'm going to show you what it looks like. You can use the same method, and we do, to engineer chestnut. This cartoon will kind of show you what it looks like. It's all out of scale and stuff, but it gives you the right idea. Here's the agrobacterium. You know, it's not nearly that big compared to the plant cell. You just saw how tiny they are. There's the plasmid, the circle of DNA, and the gene you want to put in. See that yellow thing? That's the gene you want to put in. Agrobacterium injects it in there. Now that yellow gene has made its way into the nucleus. But now you got to figure out, well, how do I get this thing to make plants, right? You want plants, because if you can start with a single cell and get it to divide and then make plants from those cells, which a lot of plants can do, that means that every cell of every plant that comes from that original cell will carry that gene and hopefully express it, right? Okay, so that's what they do. This was done originally with tobacco, which is why this shows a tobacco plant here in 1980. And here they are growing these cells in culture. And if you give these cells certain plant growth regulators, they will make shoots, or you'll see embryos that we call somatic embryos. Heard that term before? Somatic embryogenesis. I'll show you what that looks like. And then you can grow these up into plants, and this is a genetically engineered plant. That's how it's done. Looks really simple, and in many ways it is kind of simple, but getting it to really work and having a great cell culture system that will do this, that's the hard part. You can get genes into cells easy. The hard part is getting the plants to regenerate. Okay, so how about our cell culture system? This is essentially what I just told you. If you want to engineer something, you've got to have a cell culture system that you can grow plants from single cells from. We use embryogenic cultures, and I use embryogenic cultures in my lab for all kinds of trees, ash trees, yellow poplar trees, magnolia trees, chestnut trees, oak trees. This is what we work with, and every system is different. And I will tell you this now, American chestnut is the biggest pain in the ass tree I've worked with in 40 years to get to do this. You'll see it in a minute. This is what somatic embryos look like. Of course, when I got here in 1984, I said, oh, I'd like to work on yellow poplar. And for some reason, yellow poplar worked like a charm. These are beautiful little yellow poplar somatic embryos. You actually start with a single embryo from a seed, and you get it to clone itself. And that's what these are. These are all copies of one embryo that was once in a seed, and you can make thousands of them, okay? It's a very powerful tool. Then you can germinate them like little seedlings, and you can grow them up. These little yellow poplar trees, they're all genetically identical because they're a clone. So this is the definition that goes with that. Somatic embryogenesis, it's a process by which we make these structures called somatic embryos. They look like the embryos that are in a seed that germinate and make a seedling, right? But they're not. They're clones, okay? They're produced asexually, not from the union of egg and sperm, but from cells that are already there, and we fool them into making embryos. These somatic embryos can germinate to make what we call somatic seedlings. They're different from regular seedlings because they're clones and they didn't come from a seed. Okay, what does this look like in American chestnut? We found out that we had to go collect chestnuts in August before they even look like chestnuts. So there you see the burrs there? That's an immature burr, and those three little white things are 3 highly immature chestnuts that we cut out of the burr. And then we cut open each of those nuts, and inside, you can barely see them here, these are seeds because each nut actually starts out with a lot of little seeds inside. Okay. Eventually one takes over and fills the nut with the big embryo that you eat, right? That's the chestnut that you eat. This is what one looks like close up. It's only 1.5 millimeters long, and we culture that on a tissue culture medium that has everything plants like to grow on. It has sugar, it has nitrogen, phosphorus, potassium, it has plant growth regulators, it has vitamins, and if we're lucky, that seed, instead of just popping out one embryo, will pop out a cluster of embryos. These are the first somatic embryos, and if we keep transferring it to fresh medium about every month, it'll make more and more of these things until we've established an embryogenic culture. And so now we started with one embryo and we probably got about 500 there. These things will actually start out as single cells. That's what they're doing. Each embryo has cells on it and it makes more embryos, okay? You can imagine the multiplying power of this. Okay, so far so good. We can start embryogenic cultures, we can grow embryogenic cultures. This is only about 3 years of work to get this to work. How do we make trees from them? We found out the best way to grow them is not on plates of gelled medium, but in liquid medium. Same medium, no gelling agent. So those are flasks on a shaker, shaking 24 hours a day, and they're growing these cultures. What we do is we dump these cultures through some screens to get just the size cell clusters we want, and you're looking up here through the bottom of this flask. All these things that look like little light bulbs, those are somatic embryos, and they're nice and singular. They're not all stuck together, which was a problem with chestnut. All the embryos would be stuck together, and you try and separate them, you break them and damage them. And we can dump them out, and they'll start making embryos with little cotyledons, just like a seed embryo. We pick those, grit them out. They go into a refrigerator for about 15 weeks. Why do we do that? You may know that most tree seeds of our trees that grow around here require a cold treatment before they'll germinate. That's why they sit on the forest floor all winter. They're getting their cold treatment and waiting for it to get warm, and then they'll germinate. Turns out chestnut somatic embryos need the same thing that chestnut regular embryos in the nuts need. Sometimes they'll actually germinate in the cold. We get those plantlets out, put them in these things, they're called magenta boxes because they're made by the Magenta Corporation, and they grow up, they put down roots, they put up shoots because somatic embryos have both a root pole and a shoot pole, and eventually we can get them out, harden them off, and they go out to the greenhouse. That's about 10 years of work there. Where was the bottleneck? We could not get plants to save our lives. We didn't know what to do. It turned out we needed to give them a long cold treatment and some other things to get them to germinate. The reason this medium is black here, it's gelled medium, it's got activated charcoal in it because the roots like not to be in the light. They turn brown in the light. So the charcoal keeps them from turning brown. Before I show you the genetic engineering, I do want to tell you one great thing about these cultures is you can, once you establish them, store them in liquid nitrogen, and you can hold them forever. So you can conserve that germplasm forever, as long as you don't run out of liquid nitrogen. We get a giant tank of liquid nitrogen delivered about once a week to keep our cryo freezers going. This is Paul Montello in my lab. All these boxes here in this stack are filled with tiny little vials with cryo-stored chestnut material, and our other cultures too. When they come out of cryo, they look like hell. I mean, if you were stored at minus 196, you wouldn't look so good either. But within a week, they start regrowing, and eventually you have your culture back. I'd say about 85% of stuff we stick into cryo, certainly chestnuts, will regrow when we pull it out, whether it's 1 week later or 15 years later, it will come back. Okay, how about the genetic engineering? Here's the first plasmid we worked with, and I got to tell you that before we tried any blight resistance candidate genes, we had to do something to show that, oh yeah, we can engineer this. And the easiest way to do this is with something called a reporter gene. It has nothing to do with chestnut blight, but it's very easy to tell which of your embryos and cells got the gene because they turn blue, okay? So in order to do the genetic engineering, we use this Pcambia vector. It's also got an antibiotic resistance gene in it. Why is that? You'll see in a minute. That's how we select those clusters of cells that got this plasmid, okay? So what's it look like when we do it? There's our embryogenic suspension culture. Infect it with agrobacterium. This is called the selection medium. It's the same medium, but it has an antibiotic that keeps cells that do not have that NPT2 gene from growing. So they're turning brown, but you see these bright yellow clusters in here? They got the antibiotic resistance gene, and when we pull some of those out and stain for Gus, they turn blue. Okay? Then we'll pick other yellow clusters that we didn't stain blue because it kills them, and we pick the other yellow clusters and grow them up individually in flasks. If we decide to stain those, or the embryos that come from them, they turn blue. And I don't have a photo of this because I don't like it, but if we took leaves off those plants and stained them for Gus, they'd turn blue as well. So this is another 10 years of work to get this to work, okay? Well, now we're ready to try some blight resistance genes, but I want to show you one thing first, because it's going to come up at the end. This is an anther on one of our regenerated transgenic trees in the greenhouse. Do you see it's blue? It's positive for Gus. That means that the pollen that's going to come out of these anthers carries the transgenes that we put in. That means that once you genetically engineer a tree, grow it up, let it flower, the pollen flies away, you can no longer control that transgene. It's out in the general population. If it turns out to be horribly bad, too bad, it's gone now. Nothing you can do about it. You can see why some people are concerned about transgenic trees. We'll come back to that. Okay, so where do we get genes from? We got genes from all over. A lot of the genes came from Chinese chestnut. Chinese chestnut's resistant. Oh, here's a gene that looks like a good choice. So we got a bunch of Chinese chestnut genes and stuck those straight in. We also got genes from an orchid, from a grape, from pepper. And these are all genes that other people had published had antifungal products. So that's why we tested them. We thought, oh, maybe this will work on chestnut blight. But look at that one I've highlighted there. The oxalate oxidase gene. Does that sound familiar? What was the chemical that the blight fungus used to kill tissue in the tree? Oxalic acid. Oxalate oxidase, it breaks down oxalic acid. Okay? I thought I had another slide there. Maybe I flip-flopped them. This was not my idea. This was a guy named Bill Powell who knew this gene was out there. He works at SUNY ESF. He just recently retired. He's the one that said, oh, if we stick this gene in chestnut, maybe it can break down the oxalic acid before it kills the tissue in the chestnut canker. It was his idea, certainly not mine. And he started working on genetic engineering of chestnut trees himself in his own lab. These are some of our transgenic trees, and we planted bunches of them out at Whitehall Forest. Some of you know where that is, at the far end of South Millage. We have a fenced area there. We call it the nursery, but it's not used as a nursery anymore. And we planted a couple hundred trees with different genes in them. The only one that showed any promise was oxalate oxidase. We got this gene from Bill Powell. Unfortunately, you'll see on the next slide, it was driven by a very weak promoter, so it didn't express very highly. But it did make big, ugly cankers. That's a sign of resistance. But these trees still didn't survive. It wasn't strong enough to help them. And eventually, we ended the test. This is all very tightly controlled by the government. The fence has to be 10 feet tall with a locking gate, and we have to very closely monitor the transgenic trees, make sure they don't flower. It was a pain in the ass, but we did it, and now I'm not doing it anymore because I don't have time. There's the plasmid that was used. There's the oxalate oxidase gene. We still have the NPT2 gene because we got to still get the cultures out and the plants out. And so this was on there, and it says the same things I just told you. The oxalate oxidase gene is from wheat. It breaks down oxalic acid. Okay. This was what Bill got. He switched out the promoter that wasn't so good for one that was very strongly expressed and on all the time in the trees. Here's his results. This is American chestnut inoculated with blight fungus strain EP155, and it's already making a sunken, killing canker. That tree's a goner. This is Chinese chestnut, which should be resistant, and it is. It's starting to wall off that canker, and it's got a canker, but it's gonna live. Here's his oxalate oxidase tree. The wound is already healing up where he inoculated it. Why is it called the Darling 58 tree? It's called that because the president of the New York chapter of the American Chestnut Foundation, his name is Herb Darling. And Herb Darling supplied Bill's lab with thousands and thousands of dollars to do this research. So he named this tree, Bill named this tree after Herb. Herb's a very nice guy. Here's a gene that actually looks like it's going to do something. There's been more testing of this by both Bill and the American Chestnut Foundation, and it does hold up, although there are some inconsistencies, and they haven't figured out yet. But in general, it looks like it's going to be a way to get some blight-resistant chestnut trees out. But I told you all this stuff is very tightly regulated. You probably already know that. And it's regulated by the federal government, by 3 different agencies. USDA APHIS, Animal and Plant Health Inspection Service. You probably run into them at the airport when you come back from an international trip. They want to know if are you bringing any fruit in with you from Portugal? And you got to tell them, right? Or they arrest you. No, they don't arrest you. They say, give it, hand it over. They also regulate transgenic plants, crop plants, forest trees, anything. EPA, Environmental Protection Agency. Why are they regulating this? They think oxalate oxidase should be called a pesticide. They regulate pesticides, okay? And how about the FDA? Why are they involved? Food and Drug Administration. People eat chestnuts, right? So they're all involved and they all regulate this stuff. What are people worried about? These are some things that have been debated and there's publications on it, and some testing has been done too to find out, are they really concerns? There is this idea that once you engineer American chestnut with some unknown gene, that it's going to get out of control. It's going to become a weed that we can't get rid of, and it'll crowd out all the other trees, and it'll be horrible. It's not really, because it's just going to be kind of like the tree that was already there before blight came through. The oxalate oxidase gene doesn't kill the fungus even. It just makes it so it can't kill the tree. Genetic diversity. If you only use one clone, and this is something I'm concerned about, you've put everything through a bottleneck. And that Darling 58 tree is from a single New York tree that was genetically engineered, and there's some problems with that. Transgene escape. Remember, pollen's going to fly away. What if that oxalate oxidase gene turns out perhaps, that's the last dot there, to make people allergic, that they're allergic to the product of that gene, and it's out there? That would be a problem. That gene and the product it makes, oxalate oxidase, is in bread. Most of us who eat bread have never even known it's there, but it's in everything. Impact on non-target organisms. What if it kills fungi in the soil that are good, like mycorrhizal fungi that are good for the tree? This has actually been tested in tests by Bill with funding from the federal government, and pretty much they saw no effect on mycorrhizal fungi. Resistant biotype development. There is the idea that there will be strains of the fungus that mutate and overcome this resistance. That's a pretty good argument, and there's ideas about how to deal with it that I can't go into at this time, but you can pyramid other genes, and it might not be a problem because it doesn't really select against the fungus. All it does is keep it from killing chestnut trees. The fungus will still grow on chestnut trees. It also grows on oak trees and other trees where it's a saprophyte. It's not a pathogen. So you're not really selecting against the fungus that hard. Anyway, no one's going to get these trees to plant until these 3 federal agencies review everything and approve it. This is the American Chestnut Foundation webpage, where during the review by USDA APHIS of these genes, they had 2 public comment periods where they advertised, SUNY ESF and TACF want to release these transgenic chestnut trees. Read the environmental impact statement and send us your comments by email. Well, no, actually, you record them right on the site. They've had 2 of these comment periods. As you might expect, there's a lot of people that go, oh, I can't wait to get transgenic chestnuts. There's a lot of people that say, no, this is dangerous. And at this time, they have still not given the tree non-regulated status. So it cannot be released. You can grow it in field tests with approval and all the precautions, but it can't be distributed to people to grow. They had the idea that this tree would be approved, the Darling 58 tree would be approved to plant out, and its progeny to plant out by the end of this year. The TACF is not nearly as confident about that now. There seem to be stalling. Something's going on at the agency. The real problem is going to be EPA. EPA says, oh yeah, we're considering deregulating it, but if we do, it'll just be a partial deregulation for growing by certain people. It'll probably be at least another 3 years before we fully release it, if then. So that's the one they're most worried about now, is the EPA. Nevertheless, we've gone ahead and we want to grow Georgia trees that have this gene in it, right? One way to do that is to take pollen, because some of these Darling 58 trees are big enough now that they're flowering, and in fact, you can get them to flower in like a year if you want to, and make pollen by growing them under very high light intensity. So they're distributing this pollen. In 2 summers ago, we went out and pollinated some of our Georgia trees with that pollen, collected the nuts, and those trees are now being tested to see if they inherited the genes and if they are expressing them and have some blight resistance. Not by us, they're tested by the American Chestnut Foundation National. But that's some friends of mine up in a little cherry picker doing the pollinations. That was a lot of fun. And this is what I'm doing now. This is where I am. And I don't want to spend too much time on this, but okay, all this pollen is derived from this one tree in New York, right? I'm not so worried about oxalate oxidase. I'm worried that those genes around the oxalate oxidase gene in the New York tree, in the Darling tree, are going to be stuck in a big block because they're highly select. That piece of DNA, that piece of chromosome is highly selected for. So they're like, what do you call them? Not trespassers, they're hangers-on to the oxalate oxidase gene. These are genes that are adapted, making that tree adapted to grow in New York, not Georgia. They may also have some genes that aren't that great, that make it susceptible to something else. I'm uncomfortable with the fact that if we pollinate every chestnut tree with Darling 58 pollen, some piece of that New York tree is going to be in every tree and all its progeny from here on out. So and I flapped my yap, so that's why I got this grant from them. I wasn't intending to get a grant, but TACF has given me a grant to stick oxalate oxidase straight into American chestnut trees from throughout the range. These are Georgia cultures I started to engineer directly with, I didn't start them, my technician started them, to engineer with the oxalate oxidase gene directly, and maybe even with some different promoters. That gene's on all the time in Darling 58. Maybe we should just make it turn on when the tree is wounded, so that gene's not on all the time. And there's some discussions going on about this too. So these are all Georgia. There are some trees in Georgia that get big enough they make nuts, and we started cultures from them, and now we're doing engineering on those. Here's the last part, and this is a new slide. I started thinking about other things here. One is, there's people who say, I like the Appalachian forests the way they are. I like the oaks and the hickories. It's an oak hickory forest. What's going to happen to those trees? Well, if this thing is really successful, in maybe 50, 100 years, some of those chestnut trees are going to be pretty darn big. They're going to be spreading like crazy, because that's what they used to do. They used to dominate on the ridges and benches in the Appalachians. There's some estimates that 1 out of 4 trees in the Appalachians was an American chestnut. Those oaks and hickories were the subdominants then, and the chestnuts were in the top. Yellow poplars got as big as they did too. Those trees will be returned to subdominant status, and it will change the whole look of the Appalachian forests. Do we really want that? Some people like the forest the way it is now. It's what they grew up with. Something to think about. And then there's this. And this is something I was kind of unaware of. I knew Bill Powell was working with some of the indigenous nations in New York, and they were kind of enthusiastic about, oh, the chestnut tree is coming back. But there's been some more thinking about it now. The Eastern Band of Cherokees, they're kind of enthusiastic about it. But there are different tribes and different people in the tribes that say, oh, well, it's wonderful you're going to bring back this tree, but you know what? It's not going to restore all the rights we used to have to the land and the things that grow on it. And there's an article, this was in Grist, a friend of mine sent this to me, and it kind of opened my eyes. The Native American tribes in New York, their treaties, they gave up land, but they did not give up access to fishing and hunting and collecting nut crops on that land. And so now, oh, the chestnut's coming back, but no one's thinking about that, we really should have access to all that. So there's people, Native Americans are concerned about that. It sounds so great, the technology sounds so great, and some of them are not super happy that it's genetically engineered, and they'd rather have chestnuts that, were resistant from some other approach. But it's something to think about. If you're interested in this article, it's certainly got a slant to it, of course, but it's a different viewpoint that's worth looking at. Okay, almost done. I don't even look at my time. Is my time horrible or?
No, you're right. It's 6:30. Right?
Okay, so and we'll hopefully have some time for questions. These are the people in the lab that actually do the work while I sit on my butt in front of the computer. Ryan Tull is the guy that starts all the chestnut cultures, maintains the chestnut cultures, and he's doing some of the genetic engineering. Heather Gladfelter's a postdoc that is a transformation expert. She worked for biotech companies that did tree transformation, one called Arborgen. Paul Montello, he's the guy that does the cryo. Skye was an undergraduate in ecology and genetics that did some of the chestnut work as a Curo project in the lab, and I do have Curo students and other undergraduate researchers that come in the lab. Ryan McNeil works with chestnut. He's actually the director of the UGA Hort Farm, but he's also a PhD student, and he's one of the guys that was organized the pollinating out there at the Hort Farm that I showed you. And then finally, there's money, because this is expensive research, and I'm constantly looking for support. And luckily, I've had some really good supporters, like the American Chestnut Foundation. Over the last 5 years or so, has been giving me some nice funding, what they can afford. They have a lot of expenses of their own. Foundation for the Carolinas is a kind of a feeder, a pass-through organization that the guy who is the Foundation for the Carolinas is TACF's biggest contributor. So he actually designates a little bit of funding directly to come to my lab. The Georgia chapter, they help us a lot of the TAACF. Arborgen has funded our, that's a company, tree biotech company, has funded our research in the past, not now. Forest Health Initiative, they had lots of millions of dollars to work with, and this fit right into their objectives. And then a few other ones. And then I do work with the American Chestnut Cooperators Foundation too. So they've all been really helpful. And that's the end. Okay. Thank you for your patience. Thank you.